Next, we're going to look at how distant tissues within your body communicate with each other. There's two ways that they might do this. It could be through the endocrine system, or it could be through the nervous system. We will get to the nervous system and we'll talk about that in great detail. Be patient. But in the meantime, let's take a look at the endocrine system and the happy little glands that comprise it. The nervous system and endocrine system act together to coordinate functions throughout the body and to help maintain homeostasis. The nervous system relies on nerve impulses or action potentials that can rapidly move along neurons. It also relies on neurotransmitters that can be secreted into synapses, the spaces between nerve cells, or into other gaps between nerve cells and glands or nerve cells and muscle cells. The nervous system brings about very quick and very brief effects. And also the nervous system will act on very specific targets. On the other hand, the endocrine system relies on hormones. These are molecules that are released into the blood, and they travel throughout the entire body. Any cell anywhere in the body that has a receptor for that hormone is capable of responding. The responses are slower, the effects last longer, the hormone has to be removed for the effect to be removed, but there's a broader influence. The endocrine system gets its name from the family of glands that release hormones. We have two broad categories of glands. We have exocrine glands, which are going to secrete materials onto the surface of either a cavity or the surface of the body itself. They carry this material down ducts to the surface. Exo means outside, so the material is going to the outside of the tissue. Examples would be sweat glands, where sweat is released onto the surface of the skin through a sweat duct. Salivary glands, where saliva is produced within glands on the face, the parotid gland, etc., and that saliva moves through channels, through ducts, into the cavity of the mouth. Mammary glands, of course, milk is released to the outside world. Ceruminous glands, which produce earwax. Lacrimal glands, which produce tears. Sebaceous glands that produce oil that is going to be extruded onto the surface of the skin. Mucus glands that produce mucus that's going to be secreted into the digestive tract or into the respiratory tract. Endocrine glands don't have ducts. Endo means inside. Instead, they secrete their products into the interstitial fluid, the fluid that bathes the cells, the fluid that's found in between the cells. And then that material will diffuse into the blood and be carried throughout the body. You have a lot of endocrine glands in your body. This is a complex system with lots of complex interactions. Some of these glands function as master glands. These are known as tropic glands. They're glands that turn on or turn off other endocrine glands. So examples would be the pituitary, the thyroid, the parathyroid, the adrenal, the pineal, and the hypothalamus. And we'll talk about each of these in detail. Other endocrine glands don't act on each other. Instead, they act directly on tissues. And in some cases, tissues have endocrine function. And some of them you might not expect. Some of them are fairly recently discovered endocrine functions. Heart tissue, the muscle that's found within the atria of the heart, has been recently discovered to produce hormones that help regulate blood pressure. So these non-tropic endocrine glands include the thymus, the pancreas, the ovaries, testes, adrenal, etc., etc. And we'll talk about each of these in turn. Hormones will only bind to specific target receptors. They do this through what's called a lock and key mechanism. So the hormone has a certain shape, it's the key, and it can only fit in a certain lock. That would be the receptor. So the hormone needs to fit into a cleft on a receptor that matches its shape. And if that doesn't happen, then there's not going to be any effect on the cell. The hormone simply won't bind to that receptor and it won't bring about any changes. Hormones and other signaling molecules can work in a few different ways. One example is what's called paracrine signaling. And in this type of signaling, we have cells that are releasing a signal molecule, a hormone, for example, that is going to have effects in the cells in its immediate neighborhood. That signaling molecule isn't going to travel very far. A nice example of this would be damage remodeling. So let's say that you have a cut in your finger. What's going to happen is there will be increased blood flow to the area, and that will bring platelets into the area. The platelets will release a signaling molecule, platelet-derived growth factor, that will tell the cells in that area to divide and replace the damaged cells. The damaged cells themselves will also release these paracrine signaling molecules that tell the cells around them that they need to divide to repair the damage and replace the damaged cells. Autocrine signaling is not nearly as common. This is where a cell releases a compound that stimulates the cell that released it. So this can be an example of positive feedback. Endocrine signaling is what we're going to focus on. So in endocrine signaling, a hormone is dumped into the interstitial fluid, that is passed into the blood, and it gets essentially everywhere in the body. Hormones can be water-soluble or lipid-soluble, and this is going to determine how they're transported through the body and also how they interact with their target cells. The water-soluble hormones are amines in most cases. These are molecules that have carbon to nitrogen bonds within them. Remember that nitrogen is quite highly electronegative. It's going to pull electrons away from carbons, and that's going to give us an uneven charge distribution, and it's going to make the molecule water soluble. We can have water soluble hormones that are proteins or that are peptides. Peptides are short proteins, basically. We're dealing with a molecule that consists of a short chain of amino acids. The lipid soluble hormones are steroids. They contain these rings that are hydrophobic. Lipid-soluble hormones won't dissolve readily in the blood, so they have to stick to transport proteins. Transport proteins are amphipathic. They'll stick to this hydrophobic molecule, this steroid, at one end, and the other end will stick to the water. Peptide hormones and other water-soluble hormones don't enter the cell directly. What's going to happen is they're going to bind to a receptor protein that's embedded within the cell membrane. And this receptor protein is going to be a transmembrane protein. It completely crosses the membrane. After the hormone binds to the outside of this receptor, it's going to cause a change in the part of the protein that sticks out into the cytosol of the cell. That's going to bring about a change inside the cell. Usually what happens is this activates an enzyme or activates some sort of internal signaling molecule that's going to bring about changes inside the cell. Because peptide hormones are water soluble, they dissolve into the interstitial fluid and the blood very rapidly, so they can get to the target cell quite quickly. And as we'll see, by binding to an external receptor and using this internal messenger molecule, we can greatly amplify the response. We can have one peptide hormone bind to the outside of the cell, and that can very rapidly bring about a large change inside the cell. Lipid-soluble proteins, on the other hand, can easily cross a phospholipid membrane, and they can do that because they're hydrophobic, they're uncharged, and they don't interact with the charged phospholipid heads. So what happens with a lipid-soluble hormone is that it's released into the interstitial fluid, it passes into the blood and binds to a transport protein that acts like a chaperone to keep this molecule suspended in the blood. When it gets to the target cell, it leaves that transport protein behind, and then it crosses through the plasma membrane into the interior of the cell, typically into the interior of the nucleus as well. And then it's going to interact directly with genes contained on the DNA within the nucleus. It can turn genes on or turn genes off. Water-soluble hormones, they are dissolved directly into the water and then move directly into the blood, and when they get to a target cell, they don't cross through the membrane because they can't. Instead, they bind to a receptor on the surface, and that brings about a change within the cytoplasm of the cell. The set of reactions that bring about a change inside the cell is known as a signal transduction pathway. So this is how that usually happens. In this example here, we have a hormone, epinephrine, which is adrenaline. Epinephrine is going to bind to a receptor. And this receptor on its cytosolic side is capable of adding GTP, guanosine triphosphate, to something known as a G protein. That activates this protein. GTP can be used as a source of energy, just like ATP. This activated G protein is going to bind to a molecule known as adenyl cyclase. And this adenyl cyclase is going to act as an enzyme, take ATP, rearrange its shape to make it into something called cyclic AMP. It's basically just taken ATP and bent it into a circular structure, a ring structure. That cyclic AMP acts as a messenger, and it will activate an enzyme known as protein kinase A. This is an enzyme that phosphorylates other enzymes and other molecules. And this kinase is going to go do its work on a bunch of different molecules, and in the process shut down certain metabolic pathways and turn on other metabolic pathways. And in this case, it's going to inhibit glycogen synthesis and storage of glucose, and it's going to promote glycogen breakdown and the release of usable energy. So glucose will be released into the cell, and that can be broken down by cellular respiration. Epinephrine, also known as adrenaline, is something that increases the metabolic rate in your body and brings about fight or flight responses. So in this case, it's telling this cell, release a lot of glucose, you're going to need it. In signal transduction, we have two sets of messengers, or two messenger systems. We have the primary messenger, which is the hormone. And the hormone, remember, is not going to enter the cell. It's going to bind to a receptor protein, the part of the receptor protein that sticks out of the cell. That's going to bring about the activation of our secondary messenger system, and this messenger system acts within the cell, and it amplifies the input. So again, we have a signal molecule, a hormone, so it could be epinephrine again, binding to a receptor. That's going to activate the other end of this receptor that's embedded in the cytosol. And that receptor is now going to start adding GTP to a G protein. And notice that it's activating several G proteins. G proteins move around in the cytosol, and if they bump into this adenyl cyclase, they activate it. So we've got several of these adenyl cyclase molecules being activated. Each one of those is going to create a lot of cyclic AMP, and each one of those cyclic AMP molecules is going to bind to a different protein kinase. Each one of those protein kinase enzymes is going to add phosphates to a whole bunch of other enzymes and activate those. These enzymes will then activate other enzymes or other signaling molecules. So the point is here, we start with one hormone, one primary messenger, binding to the outside of the cell, and that very rapidly is amplified into a large response. So with each step in this secondary messenger system, we amplify the number of molecules involved. So one tiny change, just a tiny bit of hormone in the blood, can bring about a very large change inside the target cell very quickly. Steroid hormones function a little bit differently. First of all, they don't bind to a receptor on the surface of the cell. Instead, they enter into the cell and they pass directly through the plasma membrane. They're going to bind to a receptor inside the target cell. Generally, they pass into the nucleus and they bind to what's called a promoter. A promoter is an on-off switch for a gene. So you have about 30,000 different genes in the cells of your body, and they're going to be used, of course, to make protein. The set of proteins that's made within a certain cell determines the identity of that cell and determines what that cell can do. So we might have a steroid hormone that passes into the nucleus, and it will bind to some promoters and turn off the gene that's attached to it. But at the same time, it might bind to different promoters on different genes and turn them on. Now remember that when a gene is turned on, that gene is going to be used to make a messenger RNA. That messenger RNA is going to move out of the nucleus into the cytosol, and it's going to bind to a ribosome. The ribosome is going to translate that message into a protein. The protein is going to be modified, etc., etc. All of that takes a little while. So the effects of steroid hormones take a little bit longer to come about. Let's take a closer look at how promoters function. What you're seeing here is a gene, and we've got this dark blue part, which is the transcription unit. The transcription unit is the part of the gene that's going to be transcribed into messenger RNA. So this is the part that codes for protein. The promoter is just upstream of this transcription unit. So it's a sequence of DNA that can be bound by RNA polymerase. If RNA polymerase binds at the promoter, it will unzip the DNA molecule. It'll travel along the gene, unzipping it a little bit at a time as it goes. It'll read the template strand, and it will add the complementary RNA nucleotides. Now, when the RNA polymerase gets to the end of the gene, there's a sequence there that tells it to stop, fall off, quit what it's doing, and the RNA transcript, or messenger RNA, is going to be released. That messenger RNA is going to be processed, and we won't get into that, but there's some processing that still goes on. But after that's done, that messenger RNA is going to leave the nucleus and head out to the cytosol, be bound by a ribosome, and that information is going to be decoded to make protein. Now, in order for RNA polymerase to bind at the promoter, there have to be some other factors already bound, and these are known as transcription factors. If these proteins aren't there, RNA polymerase can't bind. In some cases, there might be proteins bound to the promoter that have the opposite effect. They actually block RNA polymerase from binding. And this is where steroid hormones come in, because they act as transcription factors. They can turn some genes on by making it easier for RNA polymerase to bind, or they can block the binding of RNA polymerase at the promoter and turn off that gene. Now, this is a rather scary looking diagram, but don't worry, I don't want you to remember any of the specifics. I want you to just get an understanding of how the identity of cells is set up, how cells determine what genes they're going to use and what ones they aren't. And I just want you to have an appreciation of how genes are regulated by promoters and the transcription factors that bind to them. Okay, so if we look at a heart muscle cell, in the nucleus we've got 30,000 genes. If we look at a neuron, we have the same 30,000 genes. And if we look at a skin cell, again, the same 30,000 genes. But those cells are all very, very different. Even though they contain the same information, they don't use all of that information. They kind of pick and choose which genes they're going to read. So in a heart cell, we'll have some genes that are turned on that are always turned off in a skin cell. In a neuron, we'll have some genes that are turned off that are turned on in a skin cell. So it differs from cell to cell. Different genes are turned on or off during development as well. And that's going to bring about the changes in the cell. So as you develop, you start out with cells that aren't specialized. They become more and more specialized as different sets of genes are activated or turned off. Okay, so with the promoter, we always have this sequence of DNA, TATA, and that's known as the Tata box. Again, don't worry about that. But we have this Tata box, and there's always going to be a transcription factor known as TF2D that's floating around in the nucleus, and it can bind to any promoter. So long as there's a ta-ta box in there, always will be. Now we have a bunch of other transcription factors. So you can see we have these general transcription factors. They can now bind to that TF2D, and there will also be some specific transcription factors, which aren't shown here, but there's going to be specific transcription factors that will recognize other sequences within the promoter, and they can bind as well. Now, if all of these factors are in place, then other complexes of proteins, like this big mediator protein, can bind. Now, that's not all that has to happen. RNA polymerase has not bound yet at this point. What has to happen next is co-activators and activators have to bind. So we have these sequences of DNA upstream from our coding region that are known as enhancers. Activators will bind to those enhancers. The enhancers will have different sequences for different genes, and there will be different activators in different cells. So the activators that are present will turn on certain genes. So the activators have to bind, and they will bind to co-activators, and that's going to bend the DNA over. Now, once this is all done, and we have that loop of DNA, then finally RNA polymerase can bind. Basically, all these proteins create a little slot that will match the shape of RNA, and RNA polymerase, I should say, and RNA polymerase will slot in there, and then it can begin doing its thing. Now, there's other sequences upstream on the DNA that are known as silencers, and they can be bound by repressors. So maybe all of this stuff is here, and everything's ready to go, but then a repressor comes along, and it binds to that silencer, and this whole thing falls apart. RNA polymerase never gets a chance to do anything. So you can see that if we have different repressors and different cell types, they will turn off different sets of genes. Now, when a cell divides, like a heart cell, the daughter cells that result will get all of these different proteins. So they will maintain their identity and maintain their function. Now, here's the level of detail I do expect you to know. So we start with a steroid hormone that is going to diffuse directly into the cell. It's going to pass through the plasma membrane, no receptor required. It's going to get to the nucleus, and that's where the receptor is. So we have a receptor molecule, a protein, that's floating around in the nucleus. The hormone binds to that and creates this complex. So it's a fusion of the hormone and the receptor. This complex can now bind to promoters. The correct sequence has to be present though. So typically it binds just upstream of the promoter. It's looking for a certain sequence, and once it's in place, it allows other transcription factors to bind and create this big mess of proteins that create a slot that RNA polymerase can bind to. And that's going to turn on the gene. Now, it could also be that there's sequences in a slightly different location next to the promoter that this complex will recognize, this hormone receptor slash hormone. And in those cases, it turns off the gene by blocking the binding of other transcription factors or by blocking the binding of RNA polymerase itself. But let's assume that it's turned on the gene. What's going to happen next is the gene is going to be transcribed by RNA polymerase to make messenger RNA. Messenger RNA goes out to the cytosol, finds a ribosome, and it's going to be used to make a protein. So we can have one steroid hormone that turns on several different genes. Again, this is based on whether or not a certain sequence is present in the promoter or near the promoter. And it can also turn off sets of genes as well. So a nice example would be testosterone. Testosterone is a steroid hormone. It'll diffuse into cells if the hormone receptor is present in the nucleus. Then this will bind to promoters and turn certain genes on and certain genes off. It'll turn on genes that are needed to code for other transcription factors, actually, that will turn on the genes that are needed to bring about male characteristics. Or it'll turn off certain genes that are involved in bringing about female characteristics during development. The interactions between different hormones and the interactions between hormones and other signaling molecules can be rather complex. As we've talked about already, if we're dealing with peptide hormones or protein hormones, they're going to bind to a receptor on the surface of the cell, and that's going to activate a secondary messenger system within the cell, and that amplifies our original input. Different cell types can respond quite differently to the same hormone. And that's because different cell types, although they might have the same receptor, will have different secondary messenger systems. So for instance, insulin can bind to a muscle cell because the receptor is there. It can also bind to fat cells because they have insulin receptors as well. But the receptors in those two cell types activate different secondary messenger systems that do different things in those two cells. How a target cell responds is going to depend on a number of things. First of all, is the receptor present? If it doesn't have a receptor for that hormone, nothing's going to happen to that cell. It's not going to respond. And this explains why you can dump hormone into the blood and have it travel all the way through the body, but still only have it affect certain target cells. The target cell has to have the receptor. The hormone concentration is going to be important. More hormone generally is going to lead to a faster, larger response. The abundance of the cell receptor. If we only have a few cell receptors, it's going to take a little longer for that secondary messenger system to do its thing. If you have lots of cell receptors, we get this more rapid amplification. The influence of other hormones. So hormones work together or work against each other quite often. We can have a permissive situation. This is where one hormone has to bind to a receptor first before a second hormone can bind. So if that first hormone isn't present, the second hormone, even if there's lots of it, isn't going to have any effect whatsoever. Hormones can be synergistic. That means we can have two different hormones that have a similar effect on a cell, and if they're both present, they bring about a much larger effect than would occur if we just had the one type of hormone. Hormones can be antagonistic as well. So we can have one hormone that will bind to a receptor and bring about a response inside the cell, but if there's another hormone present, it binds to a different receptor and it shuts down that secondary messenger system. So the two hormones are fighting against each other basically. Having all of these different interactions allows your body to fine-tune the response that's going to occur. Whether or not a certain hormone is secreted into the blood can also be regulated by the nervous system. So neurons can synapse with endocrine tissue, they can activate it and cause it to release more hormone, or they can inhibit it and shut down the release of hormone. Chemical changes in the blood can affect endocrine tissue, activate it or deactivate it. So changes in pH, for instance, can be quite important. Other hormones can stimulate or inhibit endocrine glands. These are hormones known as tropic hormones, and we'll talk about those in just a moment. Now remember that homeostasis is maintained within cells and within the body by negative feedback. If we have a change and things get out of whack, we have a change in the opposite direction that brings us back to our equilibrium. A good example would be the effects of the hormones insulin and glucagon on blood glucose. So if you have a lot of glucose in your blood, insulin is going to be released, and that will signal the cells throughout your body to take up that excess glucose, pull it out of the blood, and do something with it. If you don't have enough glucose, then glucagon is released, and that signals your liver to break down glycogen and release glucose into the blood and bring the sugar levels back up. There aren't very many examples of positive feedback. Positive feedback amplifies a change, and quite often the change is irreversible. So for example, childbirth, baby comes out, it's not going back in. But we do have some examples like the one I just talked about. You might remember that from the start of the course. That's caused by a hormone known as oxytocin. And we'll talk about oxytocin a bit later. Tropic hormones target other endocrine glands. So we have a hormone that's being produced by an endocrine gland that is then going to travel to another endocrine gland and activate it, cause it to release hormone, or perhaps inhibit it. So an example would be the hormones that are produced by the hypothalamus. The hypothalamus is at the base of the brain, and it releases hormones that travel a very short distance to the pituitary. The pituitary hangs under the hypothalamus. These hormones can bring about the release of additional hormones. So the pituitary will release other hormones when activated by these hormones from the hypothalamus. And what's rather interesting is the hormones that are produced by the pituitary are also tropic hormones. So they will travel through the bloodstream and activate other endocrine glands. So for instance, thyroid-stimulating hormone is produced by the pituitary. It activates the thyroid and tells the thyroid to release its own hormones. Non-tropic hormones are going to target non-endocrine cells. So the example here, epinephrine, which is adrenaline, is going to be released by the adrenal glands, which sit on top of the kidneys. It's going to activate a whole bunch of different tissues. One of the things that it does is it affects or stimulates cells within the heart that control the heart rate, and it increases the heart rate. I'm going to give a quick introduction to some of the hormones that we're going to talk about in more detail. We'll start with the tropic hormones that are produced by the pituitary gland. First of all, we have follicle-stimulating hormone, FSH, and luteinizing hormone, LH. Both of these are involved in stimulating the maturation of egg cells within the ovary. LH is also involved in the development and maintenance of a structure known as the corpus luteum, which is a structure that forms within the ovary after an egg has been released. That structure goes on to produce its own hormone that maintains the lining of the uterus and maintains pregnancy if fertilization has occurred. Now, we're not going to talk too much about these two hormones just yet. This is something we'll talk about in great detail in Biology 112 when we talk about reproduction. But note that the luteinizing hormone plays a major role in the menstrual cycle in females. Both hormones, FSH and LH, are produced in small amounts in males as well, and they stimulate the release of testosterone from the testes. Next, we have thyroid-stimulating hormone, TSH, and that's going to stimulate the thyroid to release thyroxine. And this is a hormone that kind of heightens the metabolism of cells throughout the body. We have adrenocorticotropic hormone, ACTH. This is going to stimulate the adrenal gland to release a hormone known as cortisol, which again brings about heightened activity of the cells in the body. It's something that's released during times of stress, especially. Melanostimulating hormone, MSH, plays a fairly small role in humans. It's not quite known how important it is in humans. In a lot of other mammals, it plays a very important role. It regulates pigmentation of the skin and fur on a seasonal basis. Then we have growth hormone, GH. This is going to stimulate growth during infancy. It causes increased cell division rates and increased growth rates of cells. We have antidiuretic hormone, which is going to be released by the pituitary, travel to the kidney, and tell the kidney to conserve water. Finally, we have oxytocin, which, as we've talked about, brings about childbirth. It brings about lactation, the production of milk, and it's also involved in some other behaviors. The release of oxytocin gives you a general feeling of well-being. It's sometimes referred to as the love hormone. It's something that's released during intimacy and during orgasm. Now let's take a look at some non-tropic hormones. And remember, these are hormones that don't target endocrine tissue. In the thyroid, we have two hormones being produced. We have thyroxine and we have calcitonin. Thyroxine is going to regulate metabolism. Specifically, it's going to increase metabolic rate. Calcitonin is going to inhibit the release of calcium from bone, so it's going to inhibit the activity of osteoclasts. On the back of the thyroid, we have the parathyroids, and they produce parathyroid hormone. This is going to stimulate the activity of osteoclasts and cause them to release calcium into the blood. So together, calcitonin and parathyroid hormone are going to maintain the balance of calcium in the blood. In the pancreas, we have the production of insulin and glucagon. And the two of these together are going to regulate blood sugar levels. So insulin will decrease blood sugar levels, glucagon will increase blood sugar levels. And we'll talk in quite a bit of detail about how that works. In the testes in males, we have testosterone being produced. That's going to regulate sperm cell production and also bring about sexual characteristics that we associate with males through development. In the ovaries, we have the production of estrogen and progesterone. Estrogen is going to stimulate egg maturation. It's going to bring about secondary sexual characteristics, so these female sexual characteristics. Progesterone is produced by the corpus luteum, which I talked about briefly. This is going to prepare the uterus to receive a fertilized egg, and it's going to maintain that pregnancy. The adrenal cortex is part of the adrenal gland, so the adrenal gland can be divided into two distinct tissues, the cortex and the medulla. The cortex produces epinephrine. Epinephrine is adrenaline, and this is going to stimulate fight or flight responses. It's basically going to gear up the body for an active response. In the adrenal medulla, we have glucocorticoids that are released in response to stress, and this is going to increase blood glucose levels. It decreases the immune response as well. Basically, it's just gearing up the body to act. Aldosterone regulates sodium content in the blood. That's produced by the adrenal medulla. Testosterone is produced in very small amounts in the adrenal glands in both sexes. Now, in males, this tiny little bit of testosterone isn't all that important because it's overwhelmed by the much larger amount of testosterone that's produced in the testes. But this is quite an important hormone in females. In both males and females, a small amount of testosterone is necessary for bringing about the adult body form, increasing muscle mass, libido, etc. Finally, we have the pineal gland, which produces melatonin, which is involved in sleep cycles. In a lot of other mammals, it also regulates reproductive cycles. Now we'll take a look at the structures and glands that produce hormones, and we'll start with the tropic hormones. The hypothalamus is a region at the base of the brain. What you're seeing in this diagram is a mid-sagittal section through the brain. Now there's a part of the brain known as the thalamus, and we'll look at that later when we look at the brain, but it's roughly within this area here. The hypothalamus is below the thalamus, which makes perfect sense. Hypo means below. So the hypothalamus is roughly this region in here. Now below the hypothalamus, we have a little pea-shaped structure known as the pituitary gland. It's about the size and shape of a pea, and you might remember from our skeletal labs that it sits into a little depression known as the hypophyseal fossa. That's a little depression within the sphenoid. Connecting the pituitary to the hypothalamus is a region known as the infundibulum. That's this little pedestal-like structure here. Now the pituitary gland can be divided into two distinct lobes. We have the anterior pituitary and the posterior pituitary. The anterior pituitary is here, so that's the front portion of the pituitary. It's also known as the adenohypophysis. It's probably easier to remember it as the anterior pituitary. Adeno means acorn, which isn't that helpful. The back portion of the pituitary is known as the posterior pituitary or neurohypophysis. That makes a bit more sense actually, because neuro refers to the fact that we have tropic hormones transmitted between the hypothalamus and the posterior pituitary by nerves. In this diagram, you can clearly see that the pituitary consists of two distinct lobes, the anterior and the posterior pituitary. Now, take note that the orientation of this diagram is the reverse of what you saw on the last slide. So the anterior pituitary is towards the right, the posterior pituitary is towards the left. The pituitary connects to the hypothalamus through the infundibulum, but the manner of connection is quite different for the two halves of the pituitary. So for the posterior pituitary, we have nerve cells known as neurosecretory cells. The body of those cells is located in the hypothalamus, and in the body of these cells, we have the production of vesicles that fill up with tropic hormones. Those vesicles are going to move down the axon of these nerve cells all the way down to the posterior pituitary. The axons are all bundled together into a structure known as the Hypothalamohypophyseal Tract. Now, when the vesicles get to the end of the axon, and they get to the axon terminals, the vesicles are going to dump out their contents, and those tropic hormones are going to activate the cells that make up the endocrine tissue of the pituitary. So the posterior pituitary will now release its own hormones. Now compare that to the anterior pituitary. We don't have neurosecretory cells that travel the whole length of the infundibulum in this case. Instead, we have what's known as a portal system or plexus. So a portal system is where we have a capillary bed that's connected to another capillary bed, and there's no passage through the heart in between. So imagine what normally happens in the body. We have blood that leaves the heart, it goes out to a capillary bed, the blood is recollected into veins that come back to the heart. In a portal system, we have blood that is going to be distributed into a capillary bed, recollected, and then move on to another capillary bed without passing through the heart. So we have this short little portal system that connects the hypothalamus to the anterior pituitary. We have a capillary bed within the hypothalamus that's going to collect tropic hormones from the hypothalamus. And those hormones are going to be carried down a short distance through the infundibulum down to the anterior pituitary, where they're going to seep out of the blood in a second capillary bed. They're going to activate the endocrine cells of the anterior pituitary, and once again, they're going to be stimulated to manufacture their own hormones. Let's take a look at this in a bit more detail, and we'll start with what happens in the anterior pituitary. Note that for both systems, we have neurosecretory cells. These are nerve cells that are going to secrete hormones instead of secreting neurotransmitters. But the neurosecretory cells are much shorter when it comes to the anterior pituitary. So the first thing to note is that we have blood flow that comes up from the heart, and it's going to pass through this artery into a capillary bed that's found at the base of the hypothalamus. It's going to pick up hormones, and then it's going to carry these hormones down to a second capillary bed down here. And those hormones are then going to target the endocrine cells within the anterior pituitary. Those hormones are going to get dumped into this capillary bed, and then those secondary hormones from the anterior pituitary are going to travel down through veins back to the heart and throughout the rest of the body. Now let's take a look at where those hormones come from. So those hormones come from these cells up here, and I'll just circle the cell bodies. So we have these neurosecretory cells that will form vesicles that are filled with tropic hormone, and those vesicles are going to travel down this short distance to this first capillary bed. And once they're in that capillary bed, then they will follow the blood flow. Once again, they're going to stimulate cells within the anterior pituitary. They will release their own hormones that will then leave the pituitary. Now compare that to what happens in the posterior pituitary. In the posterior pituitary, we have blood that comes in through this artery here, and it goes into this capillary bed, and this capillary bed is within the posterior pituitary. This is where the endocrine cells of the posterior pituitary will unload their hormone. And then this blood is going to leave through a vein go back towards the heart and be distributed throughout the body. But there's only one capillary bed. This is not a portal system. This is what we generally see through most of the body. And in this case, the hormones that activate the endocrine tissue of the posterior pituitary come from up in the hypothalamus, and I'll circle them here. Again, they're being generated by these neurosecretory cells, but the vesicles travel a whole lot further. They bypass this first capillary bed and they travel all the way down to the capillary bed within the posterior pituitary. And you can see that in a bit more detail on the right. So we can see one of these neurosecretory cells, the axon is going to travel through the infundibulum all the way down to the posterior pituitary, and then the tropic hormones that are released will stimulate that endocrine system directly. To reiterate, the hormones that are released by the posterior pituitary weren't manufactured there. They were manufactured in the hypothalamus. So the hypothalamus is going to make oxytocin, an antidiuretic hormone. It's going to package those into vesicles. The vesicles will move down through the axons of these cells, and they'll be held within the synaptic bulbs at the ends of the cells. When these hormones are needed, they're going to be released into the blood flow. They're going to be released into the capillary found within the posterior pituitary. So the posterior pituitary is not the site of generation of these hormones, it's a storage structure. It's also worth noting that oxytocin and ADH are not tropic hormones. They act directly on tissues that are not endocrine in nature. Now, if we compare that to the anterior pituitary gland, first of all, we do have endocrine cells within the anterior portion of the pituitary, and the anterior portion produces its own hormones, and they're all tropic hormones. So once again, we have two capillary beds. We have one up in the hypothalamus. It's going to receive tropic hormones from neurosecretory cells. Those tropic hormones travel a short distance to the second capillary bed within the anterior pituitary, where they flow into the interstitial fluid, come into contact with endocrine cells, and stimulate them to release a second set of tropic hormones that are then going to be dumped into that capillary bed and travel throughout the body. We can have multiple levels of hormone interaction. What you're seeing in this chart are the anterior pituitary hormones, the hormones that are released by the hypothalamus that regulate them, and also you're seeing the endocrine glands that are affected by anterior pituitary hormones. Now, once again, there's a lot more detail here than I expect you to be familiar with. You should be familiar with the hormones that are outlined in the terminology list at the end of this PowerPoint. Okay, let's take a look at an example though, just to see how complex things can get. And we'll look at the hormone at the top here called TRH. Now, TRH stands for thyrotropin hormone, and this is produced within the hypothalamus. It travels through the portal system to the anterior pituitary, where it stimulates those endocrine cells to produce TSH, thyroid-stimulating hormone. That thyroid-stimulating hormone is dumped into the blood, and it's going to travel to the thyroid gland, where it stimulates that gland to produce a few different hormones. Those thyroid hormones are going to travel throughout the body, and they'll have many different effects on different tissues, as we'll talk about. Now, note also that this TRH hormone can also stimulate cells within the anterior pituitary to produce prolactin, and then prolactin stimulates the breast to produce milk. But there's one other thing going on here. If we look at the hormone next to that, dopamine, which is also a neurotransmitter, it does a bunch of things, it can inhibit the cells that produce prolactin. And that's what that red bar here represents. This is inhibition. So if dopamine is being produced, it's going to shut down prolactin production. So again, we have this branching network, and we can have several levels of tropic hormones. TRH is a tropic hormone. TSH is also a tropic hormone. This is a nice overview of the hormones that are produced by the anterior and posterior pituitary. And what we're going to do now is just go through each of them. And let's start with the hormones that are produced by the anterior pituitary. Remember that the endocrine cells within the anterior pituitary are stimulated by hypothalamic hormones, hormones that are made by neurosecretory cells in the hypothalamus and travel through the hypophyseal portal system. Now, the endocrine cells within the anterior pituitary can also be regulated by negative feedback involving hormones that are in general circulation. The anterior pituitary hormones act, for the most part, as tropic hormones, although there are some that can also act directly on non-endocrine tissue. We have growth hormone, GH, that's going to stimulate growth and cell reproduction during development. We have thyroid-stimulating hormone, TSH, that's going to stimulate the secretion of thyroid hormones by the thyroid. We have follicle-stimulating hormone that is going to initiate the development of oocytes, those are egg cells, within the ovary. Within males, it's going to stimulate testosterone production in the testes. We have luteinizing hormone, LH, that is going to stimulate ovulation, the release of egg cells from the ovaries, and in males will again stimulate testosterone production. We also have prolactin that promotes milk secretion by the mammary glands in the breast. We have adrenocorticotropic hormone that is going to stimulate the release of steroid hormones by the adrenal cortex. These hormones bring about stress responses. We have melanocyte-stimulating hormone, which plays an important role in lots of vertebrates. Its role is rather poorly understood in humans. In this photograph, you can see two frogs of the same species that look quite different. And what's happened with the frog on the right is that the melanocytes have been stimulated by MSH, producing this darker coloration and helping it blend in with its surroundings a little better. So it's acting to bring about camouflage in this case. There are a number of animals, think of rabbits for instance, that change colors throughout the season. They're white during the winter to be camouflaged against snow, and then they're brown during the summer to be camouflaged against the dirt. Those seasonal changes are brought about by MSH. Growth hormone, GH, acts on the liver and stimulates it to release several of its own hormones. These hormones and GH stimulate cells throughout the body to grow more quickly and to grow larger and divide more often during development. It stimulates these cells within bone, muscle, connective tissue to take up amino acid and turn that into protein. It also stimulates fat breakdown so that more metabolites are available for this growth. So it plays a very important role during the growth of a child and teenager. Now, in some cases, the pituitary can produce too much growth hormone. In the photograph here, you're seeing Andre the Giant, a very famous wrestler on the right, in a very good movie, The Princess Bride. Definitely see it if you haven't seen it yet. Andre the Giant was, I believe, about 7 feet and 500 pounds, and this is because he had an overactive pituitary that was producing too much growth hormone. If you produce too little growth hormone during your childhood and during your teenage years, you can develop a condition known as Hypopituitary Dwarfism. Remember, hypo means below, so you're not producing enough GH. Dwarfism, in a medical sense, refers to someone that is under 4'10". Linda Hunt, a well-known actress, has this particular condition. As mentioned, if you produce too much growth hormone, then you can be of exceptionally large stature. The largest woman to have ever lived, Sandy Allen, was 7'7". She had a tumor of the pituitary. Of course, with a tumor, cells divide uncontrollably. They also tend to be a lot more active than they should be. So her pituitary was making more hormone than it should have, and that included growth hormone as she was growing. Robert Waldo is, to this day, the largest man that has ever lived. He passed away in 1940. He was 8'11", And once again, he had a condition where he produced far too much growth hormone. So his pituitary gland was overactive. I just want to point out that there are several conditions that can result in someone being of exceptionally small stature. Hypopituitary dwarfism is just one of these. In achondroplasia, there's a mutation in a gene that codes for a protein that's needed by fibroblasts to make collagen. These individuals have difficulty making cartilage and ossifying that cartilage. So remember that long bones develop first as a cartilaginous model, and then that cartilage is replaced by bone. That happens quite slowly in these individuals, and their long bones grow quite slowly. So Vern Troyer, who you might remember as Mini-Me, he's an example of someone that had achondroplasia. Primordial dwarfism is another potential cause. This one's quite rare. In this case, we have very slow rates of development and cell division throughout the entire body. So the individual that you see on the left, who I believe is the shortest woman in the world, when this picture was taken, she was 18 years old. Her proportions are normal because she has very slowed growth throughout the entire body, but you can see, of course, that she is of exceptionally small stature. Next, we'll look at thyroid-stimulating hormone, TSH. This is a tropic hormone that is released by the anterior pituitary. It travels to the thyroid gland, which sits just below your larynx over top of the trachea. At the thyroid, it's going to stimulate the production of T3 and T4. These hormones increase general rates of metabolism throughout the body. Iodine is an element that's an essential part of your diet. You don't need very much of it, but you need it to produce two of the hormones that are released by the thyroid. Thyroxine, which is T4, and also triiodothyronine, T3. So both of these hormones are built around a core that contains an iodine atom. Both of these hormones are going to control metabolic rate and cellular activity. So if these are released into the blood, they're going to increase metabolic rate and rates of growth and cell division throughout the body. The thyroid also produces a hormone known as calcitonin, and this is going to lower the levels of calcium in the blood and also cause calcium to be absorbed and stored within bone. T3 and T4 increase the base metabolic rate of cells throughout the body. They stimulate the synthesis of sodium potassium ATPase. So this is a protein that will move these ions across the cell membrane. These ions are quite important for all sorts of cellular activity. They increase the body temperature. They stimulate the production of protein. They're going to increase the breakdown of glucose and fatty acids. So we have increased cellular respiration throughout your cells. They're going to increase the breakdown of lipids, that's lipolysis. They're going to also increase the developmental rate of connective tissue and also the nervous tissue as well. And you can see that there is a negative feedback loop that's going to control the level of these hormones. So we have a hormone known as TRH that's produced within the hypothalamus. That's going to travel through the hypophyseal portal system to the anterior pituitary, where it's going to stimulate those cells to release TSH, which is also a tropic hormone. That hormone is going to travel through the blood to the thyroid and stimulate the thyroid to release T3 and T4. Once those get to high enough levels, they will travel through the blood back to the anterior pituitary and also to the hypothalamus, and they inhibit the further production of TRH and TSH. Once T3 and T4 levels drop back down, then TRH and TSH start being produced again. Thyroid disorders are not uncommon, and they fall into two categories. So we can have hyperthyroidism, where the thyroid is overactive. It's producing too much T3 and T4, and that's creating an overactive metabolism. So remember, hyper means above or more. Think of hyperactive. If you're hyperactive, you're more active than you should be. We can also have hypothyroidism, which is the opposite. In this case, the thyroid is underactive, and that results in an underactive metabolism. So you don't have enough T3 and T4, you feel very lethargic and tired, and you just don't have enough energy. Hypo means less than or below. One of the most common causes of hypothyroidism is lack of sufficient iodine in the diet. So remember that you need iodine to make T3 and T4. Even if your thyroid is perfectly healthy, if you don't have iodine, you can't make those hormones. Now, what would normally happen is iodine in the soil would be taken up by plants, and then you would get your iodine by eating those plants or eating animals that ate those plants. But in some parts of the world, there is very little iodine in the soil. People living in those regions don't get enough iodine from their diet. One way to counteract this problem is to add iodine to things that we commonly eat. In many countries, including Canada, iodine is added to salt. I should also mention that you can get iodine from other sources. Seafood is a pretty good source of iodine. A goiter is an enlargement of the thyroid, and this can be quite dramatic, as you can see with the woman on the left. Interestingly, a goiter can be a symptom of hypothyroidism or hyperthyroidism. So the woman on the left lives in an area of Africa where there's very little iodine in the diet. Now, because there's no iodine, she can't produce T3 and T4. Remember that negative feedback loop. T3 and T4 in the blood would normally shut down the production of thyroid-stimulating hormone. That's not happening, so the pituitary gland is producing a lot of TSH. That's stimulating the thyroid, and it's growing in size. The cells are dividing, and the thyroid is getting larger and larger and larger. So the logic here is that if there's not enough T3 and T4, maybe there's not enough cells. If there's more cells, we can produce more hormone. However, that's not the case because we don't have the iodine. We can also have a situation where we have expansion of the thyroid due to something like cancer. So let's say we have cancer in the thyroid, the cells are going to divide uncontrollably. Let's say we have twice as many cells, they're going to produce twice as much T3 and T4, and that will give us hyperthyroidism. So we've got a goiter, we've got an expansion of the thyroid, but that came first and it produced this hyperthyroidism condition. If you do have cancer of the thyroid, and it's not that uncommon, what they might do is give you radioactive iodine. So iodine isn't really used by the rest of your body. The thyroid will gobble up any iodine that's floating around in the blood. So that radioactive iodine will make its way to the thyroid, and the radiation will kill the cells of the thyroid. So this is actually a pretty effective way of just targeting those cells and killing only those cells. Underproduction of T3 and T4, so hypothyroidism, can generate some really unfortunate conditions if it occurs within a fetus or a child. So normally the thyroid would be producing these hormones to increase the growth rate of tissues, to increase the developmental rate of the child. One of the really important tissues that has to grow fairly quickly is the brain. So we can have underdevelopment of the brain, we can have cognitive mental deficiencies as a result. And also you can see in this individual here, we've got an unusual growth of the skull as well. It hasn't developed the way it should have. Now this is kind of a sad case because this individual here just simply wasn't getting enough iodine, and their mother wasn't getting enough iodine as they developed. So to treat this, you can just give the person the hormone that they're missing. So in many countries that suffer from low iodine in the diet, if you provide the individuals with thyroxine, T4, as tablets, or give them other forms of iodine, this can prevent this condition from happening. Now, this is a condition that historically has been called cretinism, but I would suggest that you not use that term because it's kind of a derogatory term. You might have heard of the term cretin before. It's not a nice thing to call somebody. It's interesting, there's a lot of terms in medicine that were completely acceptable at one time that are now rather nasty insults. Moron, idiot, those actually used to be medical terms that would apply to people that had a certain range of IQ. And you can probably think of several other medical terms that were once acceptable but are no longer acceptable. The hyposecretion of T3 and T4 in adults can lead to increased lethargy, so tiredness, and also an inability to think clearly, as well as a condition known as myxedema, which is swelling of the hypodermis, thickening of the skin, and a loss of elasticity in the skin, which is what you're seeing in these photographs. In Graves' disease, you produce antibodies that recognize and bind to the receptors for TSH on the thyroid. So these antibodies mimic thyroid-stimulating hormone. They stimulate the thyroid. It produces more T3 and more T4 than it should, so the result is hyperthyroidism. One of the interesting side effects of this is that it enhances the growth of connective tissue, including the fat deposits that are found behind the eyes, and that pushes the eyes out, and this is this condition known as exophthalmos. So the eyes can bulge out in an extreme fashion. Incidentally, the guy on the right here, Marty Feldman, he was a well-known actor. You probably haven't heard of him, but if you watch any classic old movies like Young Frankenstein, you might have come across him. On the posterior of the thyroid, we have 4 clusters of endocrine tissue that make up the parathyroid glands. The parathyroid glands produce parathyroid hormone, PTH, and what this does is it stimulates osteoclasts. Remember that osteoclasts are cells within bone that can break down bone and release minerals like calcium into the bloodstream. PTH is also going to enhance the reabsorption of calcium by the kidneys. So the kidneys are going to be told to hang on to calcium and recycle it. Also, it's going to tell the intestinal mucosal cells to absorb any calcium that's found in the food. We can have hyperparathyroidism, and this is where we're releasing too much of this PTH, and that's going to cause too much calcium to be drawn out of bones. Now, this can be due to tumors, just like we saw with tumors of the thyroid. If we have tumors that affect the parathyroid glands, we're going to have too many parathyroid cells, we're going to have overactive parathyroid cells, and we're going to get too much PTH. In hypoparathyroidism, we have less activity of the parathyroid glands, and this can be due to removal of the thyroid. Sometimes the thyroid has to be removed, for instance, because of cancer. This can lead to muscle twitches, convulsions, and it can lead, in extreme cases, if untreated, to respiratory paralysis and death. Now, the reason for this is that muscle cells require calcium to contract. That's something we'll talk about more next term, but if you don't have enough calcium in your bloodstream, you're also not going to have enough calcium in your muscle cells, and you're not going to be able to contract your muscles properly. PTH and calcitonin work together to maintain an appropriate level of calcium within the blood. Let's imagine a situation where we have too much calcium. If that's the case, then calcitonin is going to be released by the thyroid, and that's going to inhibit the activity of osteoclasts. So they're going to stop breaking down bone and releasing calcium into the blood. That calcium that's already in the blood will be removed. It'll be removed by muscle cells that will store it away in their smooth ER, and it will also be removed by the kidney and flushed out with the urine. So the calcium level will drop, and if it drops below a certain threshold, then the parathyroid cells are going to be stimulated, and they're going to start producing PTH. The PTH, the parathyroid hormone, is going to now stimulate osteoclasts, and the osteoclasts will start breaking down bone and releasing calcium. At the same time, the PTH will also tell the kidney to hang on to calcium, not to release it in urine. So now the level of calcium in the blood is going to increase. The PTH is also going to stimulate the kidneys to release something called calcitriol. Calcitriol is a derivative of vitamin D. And what this hormone is going to do is it's going to stimulate the increased absorption of calcium from foods. And that will also help bring up the calcium levels. Again, just to summarize, PTH is going to activate osteoclasts. It's going to cause them to break down bone and release calcium into the blood. PTH is also going to stimulate the kidney. It's going to tell the kidney to hang on to calcium, so not take calcium out of the blood and put it into urine, instead maintain it in the blood. Also, it's going to tell the kidney to activate vitamin D and convert that into calcitriol, which will help the intestines to hold on to and absorb calcium that's within the food. Let's move on to the pancreas. The pancreas sits just below the stomach, and it attaches to the duodenum, the first bit of the small intestine. It functions as an exocrine gland and also an endocrine gland. You can see that there's a duct that runs through the middle of the pancreas and connects directly to the duodenum. This duct is connected to these little sacs known as acini. And this isn't to scale, those sacs would be microscopic. But those little sacs are producing enzymes, things like trypsin. Trypsin breaks down protein, amylase, that breaks down starch, and several other enzymes, as well as some buffering solutions. That material exits into the duodenum and assists with digestion. Now, interspaced among these acini, among these sacs, are clusters of endocrine cells. These are known as islets, islets of Langerhans. These islets contain tissue that will produce hormones, insulin and glucagon. And those hormones are going to be dumped into the interstitial fluid, and then they're going to seep into capillaries and find their way into the bloodstream. Here you're seeing one of these islets, one of these islands of endocrine tissue. We have alpha cells that produce glucagon, and we have beta cells that produce insulin. And together, these two hormones are going to regulate the amount of glucose that's found in the blood. So this is a negative feedback system. And it should maintain blood sugar level at about 90 milligrams per mil. It was known as early as the late 1800s that the pancreas was responsible for regulating blood sugar levels. There were researchers in Russia that wanted to figure out what the pancreas did. So what they did was they took dogs and they removed the pancreas to see what would happen. And the dogs developed diabetes. They had elevated blood sugar level. It wasn't until the 1920s at the University of Toronto that researchers Banting and Best were able to isolate the compound that's responsible for lowering blood sugar levels. What they did was they took the pancreas out of dogs, effectively giving them diabetes, and then they took that pancreatic tissue, they isolated the islets of Langerhans, they ground that material up and liquefied it, And then using centrifugation and filtration, they separated out all the debris and took the fluid and injected that back into the dogs that were missing their pancreas. And they found that the dog's diabetes went away. The blood sugar levels dropped back down to normal. Then what they did is they took that solution and they isolated out all the different compounds that made up that solution, and there's lots of stuff in there, until they hit on insulin as being the molecule that was actually responsible for lowering the sugar level. So what does insulin do? Well, it tells the cells throughout your body to take up excess glucose that's in the blood and do something with it. So again, beta cells within the islets of Langerhans are going to create insulin when there's elevated blood glucose levels. That insulin will bind to receptors on the surface of cells. Now you can see in the diagram that this activates an insulin signaling pathway, and there's several different aspects to this. First of all, we have channels known as GLUT4 channels that can allow sugar to move across the plasma membrane. These GLUT4 channels are contained within vesicles, and the vesicles are sequestered away or stored within the interior of the cell. The vesicles are going to move to the surface when this insulin signaling pathway is activated, and they're going to make the cell permeable to glucose, so now glucose can rush in. At the same time, the activities of the enzymes involved in cellular respiration are going to be heightened. So we're going to get increased oxidation of glucose and increased production of ATP. Different cells behave in different ways. Liver cells will take up glucose and they'll use it to make glycogen, which is a storage molecule. Muscle cells can do that as well. Muscle cells will also be told to take in amino acids, and they'll use that additional ATP that they now have to synthesize proteins within muscle tissue. This is a rather important figure. It gives a nice summary of how different cell types react to glucose and insulin. So let's start with liver cells. Let's say that you've just had a big meal, you've got elevated glucose levels in your blood. That means, of course, that your pancreas is going to release insulin. You're going to have elevated insulin levels as well. The insulin will bind to receptors on the liver cells, and remember, that's going to cause these GLUT4 channels to move to the surface so that glucose can get in more quickly. Once inside, we have some reactions that are going to be inhibited by insulin, and those are shown as red arrows. And we have some pathways that are going to be activated, and these are shown by green arrows. So glucose in the presence of insulin is going to be converted into glucose 6P. Several of these molecules can be stuck together in chains to produce glycogen. Glycogen is a storage molecule. The glucose 6P can also be converted into pyruvate, and your cells can do a whole bunch of different things with pyruvate. They can turn those into amino acids, or they could convert it into something called acetyl-CoA, which is a molecule that can enter into the mitochondria and be broken down through the citric acid cycle to ultimately release ATP. That acetyl-CoA could also be used to make fatty acids. Now, again, if you don't have sugar in your bloodstream and you don't have high levels of insulin in your bloodstream, those green arrows, those green pathways, are going to be shut down and things are going to run in the other direction. So your liver, if sugar is not available, will take that acetyl-CoA that it got from fatty acids or pyruvate or whatever and convert it into something called ketones. Now, I've mentioned this before, but remember, there are essential amino acids that you need to get from your diet. There are essential fatty acids that you need to get from your diet. There are no essential carbohydrates. So if you have a diet where you're not eating any starch, you're not eating any sugar, the liver, instead of storing glycogen, is going to convert these intermediates into ketones that can be released into the bloodstream. And ketones can be used as a fuel source to replace glucose. Now, there are still some cells that absolutely have to have glucose. And your liver cells can manufacture glucose. They can take pyruvate, they can turn that into glucose 6P, they can turn that into glucose and release that. Okay, but back to the situation where we have lots of glucose. In adipocytes, the glucose is going to be taken in and it's going to be converted into glycerol and also fatty acids. And then those two things are going to be stuck together to make triglycerides. Triglycerides are a good way of storing energy long term. And of course, if you take in too much glucose on a frequent basis, the adipocytes are going to swell as they fill up with triglycerides. Note also that fatty acids and monoglycerides can move into adipocytes and add to this material that's being deposited within the cell. In muscle cells, the insulin is going to tell those cells to take up glucose and convert it into glycogen as a storage molecule, but also insulin tells muscle cells to take up amino acids, and the amino acids are going to be converted into protein in the muscle cell. If there's too much glucose in the blood, insulin is released, and that tells the cells throughout your body to take in that glucose, and so the glucose level within the blood drops. But what happens if the glucose level in the blood is too low? Well, that's where a second hormone comes in. A hormone known as glucagon is produced by alpha cells within the islets of Langerhans, and it's released if glucose levels in the blood are too low. It does a number of different things. It has major impacts on liver tissue. It's going to tell the liver to take glycogen and break it down into glucose and then release that glucose into the blood. It's also going to tell the liver to take lactic acid, which is a derivative of pyruvate, and use that to make glucose, and to use other non-carbohydrate molecules like fatty acids and amino acids to manufacture brand new glucose from scratch. It's a process known as neoglucogenesis, which literally means new glucose creation. This is going to increase the blood glucose concentration. If you're hypoglycemic, this means that you have a low blood sugar level. And generally, this is because your body is not releasing enough glucagon. Now, I should note, if you're on a very low carbohydrate diet and you test your blood, it may show up as being hypoglycemic, but you may be okay because you may have sufficient ketones to take the place of some of that glucose. So insulin and glucagon work together to maintain this dynamic equilibrium of blood glucose level. So let's take a look at how they work together. We'll start at the top here with a situation where we have high blood sugar. That's going to promote the release of insulin by the pancreas, and insulin is going to stimulate cells throughout your body to take up that glucose, to draw it out of the blood. It's also going to tell your liver to store that glucose as glycogen. Both of these are going to lower the blood sugar level. Now let's say the opposite is true. We've got low blood sugar. This is going to stimulate the pancreas to release glucagon. And that's going to stimulate the liver to take that glycogen, break it down into glucose, and dump that into the blood. And that's going to bring back our glucose levels to an equilibrium level. In this graph, you're seeing a plot of blood glucose level versus time in hours after a meal that contains some sugar. At the bottom in blue, you're seeing the normal expected curve for a healthy non-diabetic person. So we start out with a resting level, you could say, of glucose of 90 milligrams. We take in our meal. There's sugar in the meal, and sugar is very rapidly absorbed by the digestive system. It gets into the bloodstream quite quickly. So after 1 hour, we've got this peak level of blood glucose. It's going to drop after that, and it's going to drop because your body has begun to produce insulin. Your pancreas has secreted insulin into the blood. That is telling all of your cells to withdraw that glucose out of the blood. Blood glucose levels drop, and they may actually drop below that 90 milligram level. That's okay. If necessary, glucagon will be produced and that will bring things back up to our starting point. So again, that's the normal healthy curve. Now let's look at a diabetic. If you have diabetes, it means that either you're not producing enough insulin or you're producing insulin but your body is not responding to it. Either way, you're not going to be telling your cells to take in that excess glucose. So we start off with a much higher background level of glucose. And I should mention that this graph is for an individual that's not receiving supplementary insulin and isn't being treated in other ways. So we have this high level of glucose to start with because the cells don't know to take it in and do something with it. After a meal, we get this rapid increase in glucose, much higher than we might expect. And it takes a very long time for those glucose levels to drop back down to where we started. And again, that's because cells are not being told to take in this glucose. In fact, cells that are not receiving stimulus from insulin might actually be suffering from withdrawal. They might not have enough nutrients to do what they need to do. Not only are they not getting sugar, but some cell types are not getting fatty acids and amino acids, because remember, insulin tells cells to take up those things as well. Cells that are starving, despite being completely bathed in glucose, might start producing ketones, and ketones will be released as well. Now, if you don't have any sugar available to your cells and you produce ketones, not a problem. But if you do have lots of sugar and you also produce ketones, the ketones and glucose together in the blood bring about some chemical changes and pH changes. High levels of ketones and glucose at the same time result in something called ketoacidosis. And this is an acidification of the blood, which can bring about a diabetic coma. There are several scenarios that could bring about diabetes, but broadly speaking, we have two categories. We could argue for more, but I'm going to stick with the simpler, traditional categories of diabetes. We have type 1 diabetes and we have type 2 diabetes. In type 1 diabetes, we have the hyposecretion of insulin. So the person is not making enough insulin. They eat a meal, their pancreas produces some insulin perhaps, but not enough to bring glucose levels back down to where they should be. So this is referred to as insulin dependent. The condition is dependent on the amount of insulin that's present. And we can treat this by using supplemental insulin. So a person with type 1 diabetes can inject themselves with insulin just before they eat a meal. The insulin will cause that excess glucose to be taken in normally. And that's because the receptors for insulin are fine. They're just not making enough insulin. This has traditionally been referred to as juvenile onset diabetes, although that term isn't used as often. The reason that term was used is because it usually appears very early in life. It's often something that you're born with. There's an issue with the cells that produce insulin. But this can appear later as well. You can have an older individual that develops damage to the pancreas, maybe through some sort of autoimmune response. And once again, their pancreas doesn't produce enough insulin as a result. Next, we have type 2 diabetes. In these individuals, they can produce normal functional insulin, but they don't respond to it. The receptors can be mutated in some cases, or the receptors just simply have given up responding. This is referred to as late onset adult diabetes quite commonly, although again, that term isn't used as often, and that's because it's something that typically appears later in life. Imagine that you have a very high caloric diet and you take in a lot of sugar constantly. What's going to happen is the cells that have insulin receptors will simply stop responding to that input because it's a constant stimulus. You won't take in that glucose and glucose levels will remain high in the blood. Now, if you have this condition, the best way to manage it is by diet. greatly reducing your sugar intake, and also through exercise. Exercise will promote the intake of glucose and hopefully bring back some of those natural responses to insulin. Now, this doesn't necessarily have to be a late onset disorder. We could have individuals that are born with a mutation in their insulin receptors, and they'll have very similar conditions. They will not respond to insulin despite making appropriate levels of insulin. I hate to say it, but the vandalism on this billboard is kind of appropriate. Imagine if you were to consume this meal on a regular basis. Most of the calories are coming from sugar, monosaccharides and disaccharides that are taken in by the digestive system and put into the blood very quickly. That is going to produce a massive spike in insulin. And if you're doing this frequently, then your body will start to ignore that insulin signal because it's always there. Now, another thing to note is that these food items contain a fair bit of fat as well. We have lots of fat in the whipped cream and the ice cream. If you're taking in sugar and fat, sugar's easier to break down. Your body will focus on that, or at least it'll try to. It'll ignore the fat and instead push that directly into adipocytes, into fat cells. It'll go directly into triglyceride storage. So that's another side effect of eating a diet like this. Now, humans are very adaptable. We are opportunistic creatures. We can live on a variety of different diets. There have been cultures that have survived mostly on rice, although their caloric intake tends to be quite low. There have been cultures that have lived entirely on proteins and fats, like the Inuit, for instance. But a very high caloric diet that consists of a very high proportion of sugar is a bad idea. And that's the one diet I would definitely discourage you from. Let's move on to something totally different. We're going to look at the ovaries and the testes. These are gonads, of course. Gonads are organs that produce gametes, but they also produce hormones. So the ovaries produce estrogen, and there's two types, and also progesterone. And we'll talk about these in much more detail when we talk about the reproductive cycles in Biology 112. Remember that the hypothalamus produces FSH, follicle stimulating hormone, and also LH, luteinizing hormone, that help maintain the menstrual cycle, maintain pregnancy, and prepare the mammary glands for milk production. All of these hormones are also involved in the production of secondary female characteristics. We have more hormones, inhibin, that can inhibit FSH, relaxin, that is produced during pregnancy. But again, we'll come back to these next term. In the testes, we have the production of testosterone. Testosterone is going to regulate the production of sperm, but it also maintains and generates male secondary sex characteristics. I mentioned at the beginning of this topic that steroid hormones are often involved in gene regulation, turning some genes on and turning other genes off. This can be really important during development. One of the best examples of that are the actions of testosterone. So we'll spend a little bit of time talking about that. And as mentioned, during 112, next term, we'll come back to this in more detail and look at all of the sex hormones and all of the regulatory activities they're engaged in. Anyway, in the early embryo, we have a region known as the urogenital ridge, and this is going to contain cells that will develop into the gonads. We have what's known as a bipotential gonad forming first. Bipotential because it has the potential to develop into an ovary or a testes. Within the bipotential gonad, we have supporting cell precursors that will develop into support cells, cells that will nurse the developing germ cells. We have primordial germ cells. Germ cells are cells that undergo mitosis, but some of them will enter into meiosis and give rise to egg cells or ultimately to sperm cells. And then we have these steroidogenic precursors that will give rise to cells that produce steroids that bring about female characteristics or male characteristics. I have to admit, I've crammed a lot of information onto this one slide. Let's take a bit of time and break it down. And we'll start with the diagram on the left. Now, if you're female, you have two X chromosomes. You got one from your mother and one from your father. If you're male, you have one X chromosome that you got from your mother and one Y chromosome that you got from your father. The Y chromosome doesn't have a whole lot of information on it. It's got a few genes. One really important one, and that is the SRY or TDF gene. The X chromosome, on the other hand, has over 1,000 genes. But that SRY gene on the Y chromosome is responsible for bringing about male characteristics. It's also known as the TDF gene, which stands for the testes determining factor, because it brings about the creation of the testes. If the SRY or TDF gene is not present, the embryo will develop as a female. That's the default pathway. But if that gene is present, the ovaries will develop into testes instead. The testes will produce testosterone, and that brings about other changes. Okay, so that SRY or TDF gene is going to serve as a master gene. It actually codes for a protein that's a transcription factor, and that protein will turn off a whole bunch of genes, the ovarian genes, the genes that are needed to generate ovaries, and it will turn on a whole bunch of other genes that are involved in making testicles. So all of these genes are present whether you're male or female, just they're turned on or off based on whether or not that SRY gene is present. The SRY gene is also going to turn on another master gene known as SOX9. Don't worry about that at all. Just realize that SOX9 is another gene that codes for a transcription factor, and it's going to turn on more testicular genes and turn off more ovarian genes. So if this one gene, SRY, is present in the very early embryo, it is going to impact the activities of about 4,000 other genes, which is pretty amazing. So that one gene will have a big impact. Okay, let's look at the diagram at the top. So within the area that's going to give rise to the reproductive organs, we have what's known as the genital ridge. Within this area, if there's no Y chromosome, the gonad that's forming there, the bipotential gonad, will develop into an ovary. If there is a Y chromosome, that Y chromosome is going to have this SRY or TDF gene, and that is going to tell that bipotential gonad to develop into a testes. So we'll have testicles developing. They're going to have two populations of cells, Sertoli cells that will produce something called Mullerian inhibiting substance, MIS, and we'll come back to that, and also Leydig cells that produce testosterone that brings about further changes. We also have germ cells. If it's an ovary, those germ cells are going to develop into egg cells, ova, and if it's a testicle, then these germ cells are going to give rise ultimately to sperm cells. Now, one thing that's really quite interesting that I'll get to in just a second is that males and females have the same parts. We really do. The parts are just arranged a bit differently. When we develop as very early embryos, we also have the same plumbing. We have the same ducts. So we have these sex ducts. And we have two sets of ducts as early embryos. And it doesn't matter if you're going to develop into a male or a female. You have these two systems in place. There are Mullerian ducts, and the Mullerian ducts will ultimately give rise to the uterus, the fallopian tubes or oviducts, and the upper portion of the vagina. Now, if you are male, those ducts will degenerate. There are also Wolffian ducts. and they give rise to the epididymis, the vas deferens, the seminal vesicles, the ejaculatory duct. They will persist if you're male, but they will degenerate if you're female. So the diagram at the bottom there, the bottom right, shows these two ducts. We have these Mullerian ducts, which are here, and also here. They're going to persist in the female, and you can see down the bottom that they're going to give rise to the uterine tube or fallopian tube, the uterus, and the upper part of the vagina. Now in males, we have these Wolffian ducts that are shown in green. They are going to persist. They are going to break down in females. In males, they're going to give rise to the ductus deferens or vas deferens that you see here. So again, we start with the same pieces, but they develop a bit differently. In the male, in the early testes, we have the secretion of something called MIS, Mullerian inhibiting substance, and that's what causes the Mullerian ducts to break down in males. Let's also look at what's happening externally. We'll look at the development of the external genitalia. And as I mentioned, males and females start out with the same bits. At the top in the middle, you're seeing the bipotential stage. This is a 6-week-old fetus. It's neither male or female at this point. At this point in development, the fetus actually has a very pronounced tail, and that's being cut off down the bottom here in case you're wondering what that stump is. The tail has been sectioned through so that we can have a view of what's going on. Above that, we have the genitalia. We have the genital tubercle. Tubercle means a nub or projection. That's going to develop into the head or glands of the penis or into the clitoris. So those parts are analogous. They are the same. They develop in a similar way. Underneath that, we have the urethral groove. So we have this little cleft that is going to develop into the cleft between the labia and the opening of the vagina in females. And in males, it's going to seal over and it's going to give rise to this ridge here along the scrotum and the underside of the penis known as the raphe. We also have swellings on either side of the urethral groove. These swellings will develop into the labia in females. They develop into the scrotum in males. And then, of course, the gonads are going to drop down into the scrotum in males. They're going to stay put in females. So in both males and females, we start out with one opening, the cloaca, and that's going to separate out into the anus and then also into the urethral groove. And then we're going to get the development of the labia and the clitoris in females. In males, we have the same structures. The only real difference is that the glans is going to move to the tip of a shaft and the urethra is going to migrate into the glands. But otherwise, we have the same structures. They're just arranged a little differently. Take a look at the women in this photograph and try to figure out what they have in common. You'll probably be surprised to learn that they all have a Y chromosome. Genetically speaking, they're male. But of course, they're not male. What's happened here is that each one of these women has a functional Y chromosome with a functional SRY gene. However, they lack functional testosterone receptors. So the gene that codes for the testosterone receptor is not functional. So what that means is during early development, they produced the SRY protein. That caused the gonads to develop into testes. The testes produced testosterone, but there was no response. None of the cells that had receptors could respond. The default pathway for the embryo is female. So in the absence of testosterone or in the absence of a response to testosterone, the embryo will develop as female. Now it's also worth noting that the testicles themselves have testosterone receptors. Again, those weren't working in these individuals. There's a positive feedback loop that occurs. So if testosterone is present, the testes continue to develop and eventually drop down into the scrotum. Without testosterone present, the testes degenerated. This condition is known as androgen insensitivity syndrome, or AIS. Andros in Greek means manly. An androgen is a hormone that brings about male characteristics. These individuals are insensitive to those hormones because they lack functional receptors for them. Somewhat ironically, the mutation that causes this occurs on the X chromosome. That's where we find the gene for the testosterone receptor. Now remember that these individuals do develop testes, and during early development, those testes are producing testosterone, even though it's having no effect, and they're also producing MIS, Mullerian inhibitory substance. That is having an effect. That will shut down the generation of the uterus, the fallopian tubes, or oviducts, and the upper portion of the vagina. So these individuals, although in all appearances they are female, they are sterile because they lack those inner components of the female reproductive system. The woman on the right here, Eden Atwood, is an individual that has AIS, and she's rather outspoken about it. She's also quite a talented jazz singer, and you can find videos of her singing and also talking about this condition on YouTube. This has been a rather long topic. We've only got a couple more hormones to go. On top of each kidney is a gland known as the adrenal gland. Ad means towards, renal means having to do with the kidney. So they're located towards the kidney. They're actually right on top of the kidney. They're also referred to as suprarenal glands, sometimes supra meaning above. The adrenal glands can be divided into two regions. The innermost portion of the adrenal gland is the adrenal medulla, and then surrounding that we have the adrenal cortex. These are distinct regions that generate different hormones. The adrenal glands can be activated by the sympathetic nervous system, but also these tissues can be activated by ACTH, which is a hormone that's produced by the anterior pituitary. So we have these two distinct regions to the adrenal gland, and they have distinct functions and generate separate sets of hormones. Within the adrenal cortex, we have hormones being produced that are involved in regulating mineral homeostasis. We have cortisol being released. Cortisol will tell the liver to dump sugar into the blood. It'll tell cells like muscle cells to take up that sugar, and it generally heightens the activity of cells throughout the body. So it's involved in fight or flight responses and in stress responses. We also have androgens that are produced by the adrenal cortex. They don't play a big role in males because males are producing lots of testosterone from the testes, and that overwhelms the effects of these androgens. But they are quite important in females. They bring about increased development of tissues. The adrenal medulla is going to produce epinephrine and norepinephrine, also known as adrenaline and noradrenaline. These are responsible for fight or flight responses, and they enhance the effects of the sympathetic nervous system. The sympathetic nervous system and the adrenal medulla work together to bring about a number of important changes throughout the body. And that's something we'll talk about more when we talk about the autonomic nervous system. Cortisol is going to heighten a number of metabolic activities within cells. It's going to increase the rate of lipolysis. That would be lipid breakdown. It's going to increase the rate of gluconeogenesis, which is the production of sugar from pyruvate and other molecules. And also it's going to increase the rate of protein catabolism, protein breakdown. These metabolic pathways make energy available to cells. This is something that can continue for quite a long time if you're in a stressful situation. And if it does continue for too long, that can cause damage to your body. So those were the hormones of the anterior pituitary. Well, the major ones at least. Now we're going to look at the posterior pituitary. And fortunately, there are just two hormones to look at. Remember that the posterior pituitary doesn't actually synthesize hormones, it stores hormones that have been produced by neurosecretory cells within the hypothalamus. And the hormones that we're going to look at are oxytocin and antidiuretic hormone. Oxytocin is responsible for two positive feedback loops that we've talked about a few times in this course. Remember that if a baby is pushed against the cervix during the initiation of childbirth, there's going to be stretch receptors within the cervix that will stretch and send a signal via the nervous system back to the pituitary gland. The pituitary gland will release oxytocin, and that brings about contraction of the smooth involuntary muscle that makes up the wall of the uterus. The baby is going to be expelled more forcefully against the cervix. That causes more stretching, that causes more oxytocin release, etc. The final result, of course, is that the baby is going to be pushed out of the uterus. A similar thing happens with the production of milk. So a baby suckling at the nipple, again, is going to stimulate stretch receptors within the nipple. That's going to result in oxytocin being released from the pituitary. That's going to result in more milk being produced. That encourages more suckling by the baby, etc. It's also a positive feedback loop. Oxytocin can be given to induce early labor. In fact, I've had to do it a few times in my research to get eggs out of reptiles before they were quite ready. Anyway, another interesting thing about oxytocin is that it can bring about feelings of closeness and well-being during moments of intimacy. In fact, it's thought that oxytocin is released when taking the drug ecstasy. Not that I would suggest you try that, but that seems to bring about some of the feelings of closeness and friendliness that are associated with that drug. Antidiuretic hormone, ADH, is also stored within the posterior pituitary. It's involved in regulating water balance within the body and also regulating blood pressure. It acts on the kidney and tells the kidney to retain water. So a diuretic is something that causes you to urinate more. An antidiuretic is something that causes you to retain water. So the major role is its effects on the kidney, and we'll talk about how that works a bit more when we talk about the kidney next term. But it also affects sweat glands. It tells them to release less water and conserve water. And it also tells arterioles to constrict. And that means that the volume of those blood vessels is reduced, and because the same volume of blood has to be squeezed through those blood vessels, the blood pressure increases. The last hormone we're going to talk about is melatonin. Melatonin is produced by a fascinating little gland called the pineal gland. The pineal gland appears to be related to photoreceptive glands within other vertebrates. A lot of fish, for instance, have a pineal gland that is sensitive to light. Now, our pineal gland is not. It's deep in the dark recesses of the skull, but it still is used to regulate the biological clock. It regulates our circadian rhythms. So on the graph at the bottom, you can note that melatonin builds up in the body during the time that we're asleep. So when it's dark, melatonin builds up, and once it gets to its peak, this also brings about the production of cortisol. And cortisol peaks just before we awake. So this is a hormone that's going to get us up and ready for the day. It's going to heighten activity. So this coordinated falling and rising of melatonin levels and also cortisol is responsible for our daily rhythms. That was certainly a very long topic, about 2 hours in fact. So congratulations, we made it through. I want to point out that there are lots of other hormones than the ones we talked about. The endocrine system is remarkably complex, and there's lots of interactions between all of these different hormones that need to happen to maintain homeostasis. But you're only responsible for the hormones that are on this terminology list.